Hello, I'm Chris Kruger, Senior Manager for Content with the Davis Phinney Foundation, and I'm delighted to have the opportunity to be here today with Dr. Claire Henchcliffe to talk about cell therapy, gene therapy, how they differ, and how they might help people with Parkinson's to live well. It's worth noting that we're recording today on May 6, 2025, and in the recent weeks, there have been some publications about these very topics, including one publication for which Dr. Henchcliffe was a contributing author. And you'll be able to find some links to these resources in the description below this video. So if you're looking for more of that information, please check out the description. You can find it there. So Dr. Henchcliffe, thanks so much for being here. Can you maybe start by telling me a little bit about your background and how you came to be involved in Parkinson's?
Absolutely. I think, obviously I'm not from the US. I grew up in the UK. And since I was a kid, I was just really always interested in how stuff works and in particular how the brain works. So it got me into training in Oxford and Cambridge studying cell biology. And I also was pretty interested in genetics. So you can see where these things are starting to come together now, right, in terms of an interest in Parkinson's. So it was a little bit after that human neuroscience was really taking off. It was getting to be really exciting. And I went back to medical school at the time. I was sort of interested in everything. But honestly, I had the chance to spend time in Stan Farn's clinic. And for those of you who know Dr. Farn, I mean, he's just an amazing figure in this field. And it took one day, and I knew exactly where I wanted to focus. So it's really been a privilege to spend all of these years working with people with Parkinson's. And I think it's a really fascinating area because there's so many treatments. There's a rich pipeline, but we know we have a lot more to do, right? We have a lot more to learn.
Yeah, it's one of the interesting things for me. And by the way, I should say I live with Parkinson's myself. And one of the interesting things for me is that there are just so many ideas. And on the one hand, you could say, well, maybe that means we don't know a lot. But in fact, I think we do know quite a bit. And I think there's still so much to learn. So it's rather exciting to know there's so much potential. And we're going to talk about some of those potential treatments today. So maybe we can start there and maybe it's best to start with a kind of basic overview. Can you maybe provide a sort of high level 30,000-foot view of what cell therapy is, how it differs from gene therapy, and why they each might be helpful for people with Parkinson's?
Absolutely. And why they might eventually combine even.
Oh, yeah, good point. Yeah,
It's another exciting possibility. So let me start off with the cell therapy. So I think when we're talking about cell therapy for Parkinson's disease, there are kind of two broad approaches. And the one that's a real focus right now is trying to replace cells in the brain that are lost, that are damaged and lost in Parkinson's disease using cells that have been grown in the lab. So that's really what the vast majority of current clinical trials are looking at. So we can talk a bit more about that. The other possibility, though, that I don't want to miss out on, it's using cells that come from blood or bone marrow or fat. They're called mesenchymal stem cells. They're a little bit different than the other types of stem cells that we're working with. They don't replace lost cells. If you inject them, they're not really going to survive, but they can deliver beneficial factors that might be neuroprotective. So there are some clinical trials that are going on in that area. So I just described a fairly circumscribed and quite limited rationale for using cells in Parkinson's to replace those that are missing. And I think there are lots more possibilities, but those are the ones that we're looking at right now. Gene therapy is kind of interesting because people have taken a lot more different approaches, different rationales. So rather than trying to replace cells with more cells, what you're doing in gene therapy is you're taking DNA, the genetic material, you're engineering it for delivery to make whatever you think you want to improve a person's brain cells in the case of Parkinson's disease. Now, DNA won't get into brain cells on its own easily, or other cells for that matter. So the gene therapy takes this trick that is really smart. There are certain viruses that will infect brain cells. So you can take those, you can kind of cripple them, if you like. You can use them like a Trojan horse, pack them with the DNA that you want to get into the cells, and then let the viral vector sort of do your work for you. So these are kind of non-infectious versions of the viruses, viral vectors. They're engineered so they'll get entry into particular cells, delivering the DNA that you have packaged into them. They can't infect and they can't replicate. It's sort of like a Trojan horse. So it's sort of disguising the DNA that you want to get in. And we can talk about some of those strategies, but rather than thinking about, replacing dopamine inputs, gene therapy gives you the ability to do that for sure. But you can also deliver other genes. You can think about correcting genetic mutations or risk factors that contribute to Parkinson's. You can think about getting in neurotrophic factors that are protective. Or even, this is a little more audacious, but it's been done, trying to reprogram a whole network by addressing one specific nucleus within the brain. So lots of overlaps there, but definitely some differences that we can see even at this early stage in terms of what's possible.
Great. Yeah. So thanks for that. There's so many questions that can come out of what you just said, and I guess I want to see if we can maybe dive a little deeper into gene therapy first, because that's where you were just discussing some of the strategies. I guess can you say a little bit more about, you mentioned you can increase, and I think you said you can increase the presence or the efficacy of certain growth factors or something to that effect. Can you say more about that? Did I get that right?
Yes, absolutely. These are neurotrophic factors. So when I think about gene therapy, I guess there are a couple of ways that I think about it. One is the type of gene therapies that have been done, and these tended to be the earlier clinical trials where people were trying to improve symptoms. So you deliver genes that would help the cells reprogram cells so they would make more dopamine. That would be one approach. Or the other approach was to modulate a network that has gone wrong in Parkinson's disease and kind of get it back to some sort of pseudo normality. So the whole idea there was, you're going to decrease your off time, or if you're off and your medicines aren't working, your off time is going to be better. So those are the initial ideas. And then I think more recently, it's been really interesting. So you mentioned the protective factors. The first part of the program was looking at a protein called nurturin, which is in the same family of proteins as the one that's being currently looked at, GDNF. So this is glial cell-derived neurotrophic factor. And this does all sorts of really great stuff to cells in culture and to preclinical models of Parkinson's disease. So it kind of helps your cells survive. It protects them against insults. It helps them talk to each other better. It helps them function better. And so the rationale is if we could get the GDNF into the brain and help cells that are under threat from the Parkinson's pathogenesis, all the things that we know go wrong, the neuroinflammation, the energy metabolism, all of this. If we could get the GDNF in there through a gene therapy, that's got the potential to do a couple of things. I think it's got the potential to help symptoms, because if there are cells that are not functioning well, the GDNF could boost their function. And then it could also help in the long term, because you're sort of protecting and perhaps restoring cells and cell function. So that's been a really interesting one, waiting to see results come out in a peer-reviewed publication. But we've seen some initial encouragement, I think. And then the last the last strategy that I think is really, really interesting for a subset of people with Parkinson's. So we know that a certain number, a certain proportion, although it's low, have a genetic form of Parkinson's. And it may be one gene where either you have a mutation that's causing the Parkinson's, or there's a change, a different allele of the gene, a different version of the gene that is a risk factor. And there's a gene called GBA1, which accounts for some people's Parkinson's. And depending on the nature of the change in the GBA1, it can actually be a more aggressive form of Parkinson's with more cognitive problems. Not for everyone with the GBA1, but for some. So I think that makes it a really compelling version of Parkinson's, where there's a clear need for something much better. And the idea there is if the GBA1 change or allele risk factor is causing less of the protein glucocerebrosidase to be made, why can't you simply put in a normal, optimized, highly functioning GBA1 gene. And then your own brain is going to make more of that protein. And in theory, it should take away the progression of Parkinson's disease. It should just restore a lot of the function and help to take away that progression over time. So I think for me, those are the really interesting areas of gene therapy right now. And then, of course, down the line, we're going to see some different approaches, right? Everything right now takes either a surgery or some sort of needle into to introduce the gene therapy into usually the brain tissue itself, although there's been an example of introducing it into the cerebrospinal fluid. But wouldn't it be great if you didn't have to do that, if you could deliver it intravenously? And that means you've got to be able to get it past the blood-brain barrier. So that's where a lot of advances are getting looked at right now, whether it's mechanically doing that or whether it's altering that Trojan horse, that viral vector, so that it can get through.
Yeah, I mean, so thanks for that. That's you just gave a lot of information about a very complicated subject, and so I'm going to ask you to do a little more if you're willing. Is that okay?
Absolutely.
I just wanted to see if we can clarify. There are a couple. GBA is not the only gene that could be said to be causal
Right.
of Parkinson's. Is that true?
Correct. I when I was training, Parkinson's is not thought to be a genetic disorder at all. And now, of course, we know that's completely untrue. And it's got really complicated genetics. So there's a whole number of genes that, if you have a mutation in even just one copy of the gene, that can lead to Parkinson's disease, like LRRK2. There are other genes where if you need mutations in two copies of the gene, and that's going to give you Parkinson's, or at least a high chance of getting Parkinson's, let's say like Parkin. And then we've got a whole bunch of genes where if there are certain changes, they're not going to give you Parkinson's for sure, but they're going to increase your risk. And so I think that we've got a few different ways that we can start looking at those. So GBA1, I think, is a really great test case, but there are many others, many other areas, including LRRK2, Parkin, PINK1 to go after.
Yeah, thanks for expanding on that a little bit. I just want to make sure that somebody who might think they'd heard something about LRRK, I didn't want somebody to listen to this and think, oh, why didn't Dr. Henchcliffe say that? And so now we've heard a couple other genes that are involved in that, and so I just want to make sure we got that covered. But I
Yeah.
also want to plug that we have some other content on our website, dpf.org, about genetic testing, genetic counseling, and also some of the environmental risk factors, which might as far as tell me if I'm right about this, but I understand that if you have one of those genes that might make it more likely you'll develop Parkinson's, or one of those variants that might make it more likely you'll develop Parkinson's, that certain environmental exposures might then push that down the road a little bit further, and then that's where you may be developing Parkinson's, head injury or toxin exposure, things of that nature.
Oh my goodness, and that is so complex. And thank you for developing content on that because I think this is an area that we love to talk to people about in our clinic. When should you get genetic testing? What does it mean? Who do you tell? What are the risks around it?
Yeah. So we do have some content on that. You'll find those links in the description, as well as the links to the studies that we talked about earlier. So maybe can you say a little bit more about the state of gene therapy in Parkinson's today? Are there any that are approved by the FDA now, or is it all in trial still? Can you say more about that?
Yeah, everything is in clinical trial. It's all experimental, and so it's incredibly tightly regulated. So if you went out and tried to get gene therapy today, no, it's not. It's not going to happen. So the approaches that are used right now, they all need a surgical delivery of the gene therapy, or the only one that doesn't is the GBA1 program, where they're actually injecting the gene therapy into the spinal fluid, actually the cysternal fluid right up next to the brain. So you can imagine, there's some risk with those procedures. I think that the second thing is that although generally it seems in the field from the many, many clinical trials that have been done so far, it seems that this is safe overall. It seems that there are either not intolerable side effects or the side effects that have been seen have generally been manageable. So we've got over that hurdle. And I think what's not so clear yet is how efficacious this is going to be. And I would just say, at least with some of the early programs, the initial ideas around trying to increase dopamine production, um, you can see some signs that it was working in some cases, but overall, the data just wasn't there to be able to push it forward. And so I think that when people go into a clinical trial, one of the big things that we have to discuss when we're taking informed consent from them is, this balance of risks and potential benefits. And I think in these early trials, you know what some of the risks are. There are some risks that are likely unknown. And, there have been examples of unpredictable risks. And then when you get to the efficacy, you know what these interventions can do in preclinical testing, but until you get into humans, it is really hard to tell. And there's so much that goes into that. Are you providing gene therapy to the right people at the right time? Are you too late? Is the process already done with, and there's nothing you can do about it? So there's so much in there, and that's why I think it's there's a lot of enthusiasm in this field. There's a huge amount of development of new technologies, and that really gives me a lot of optimism. But just right now, you can't hang your hat on having a single efficacious gene therapy.
Well, thanks for sharing that information, for clarifying some of the risks involved. And I'm gonna take advantage of my position right now, and I'm gonna say I would never suggest anyone participate in any particular clinical trial. I don't think that's for me to say, but I do think what you just said about, yeah, the fact that you might be too late with a certain intervention is a reason for someone to think it would be a generous act for perhaps for them to choose to participate in clinical research of any type. Now, that's not to say I'm not encouraging anybody to participate in any particular trial, but it is a reason to participate in trials generally, that you can help advance treatments more quickly. The more people enroll, the more quickly trials get executed, the more likely we'll be able to develop new treatments that can help people before it gets to be too late for them to have an effective treatment when efficacy is proven for any given number of treatments.
And thank you for saying that. And I think it's also important to realize that we can, as researchers and investigators, we can argue all we like about when is the right time, when is it too early, when is it too late. But in the end, until you do it, you don't know,
Mhm.
right? So I think your point is really well taken.
Thank you. So maybe let's pivot to cell therapy, should we? And maybe can
Absolutely.
you just describe some of the strategies and of different cell therapies and what the benefits and costs are here?
Yeah, absolutely. So I think, first of all, the idea of using cell therapy, it's not new at all. I mean, this has been an idea kicking around for decades. I think what is new, it's the advances in stem cell technologies, and that's what we're really excited about. This ability to grow almost unlimited numbers of cells in the lab, so you can truly do all the testing that's needed to properly regulate, to properly understand what those cells are going to do, and have, cell banks where you can draw for testing on the one hand from that cell bank, but then you can take cells from the same cell bank that are actually going to be for your intervention. So I'll just say, I mentioned already there are different types of stem cells. The ones that I mentioned, the mesenchymal stem cells that come out of blood and bone marrow and fat, I'm not really going to be talking about those because those are less investigated. I think they're still valuable, but really the most exciting results recently, I would say, have come out of either using cells that have been grown in the lab from human embryonic stem cells. So those are unmatched. I wouldn't call it a one size fits all, but at the same time, it's like a one cell line that can be used for multiple different people. And then, I'll just say a little bit about the human embryonic stem cells. They're grown originally from extremely early stage human embryos, when the embryos are just a few cells. The subject to very, very stringent ethical and regulatory requirements, obviously. And once you get those few cells, you can grow them in unlimited quantities, basically, in the lab. And so, for example, cell lines that I've worked with, they've been grown in the lab since, well, I don't know, but for decades, basically. And being able to do that means you can learn a lot about the cells. You can really understand them very well. It's more like a standardized approach. And then, and John Gurdon and Dr. Yamanaka got the Nobel Prize for this back, I think it was 2007. They discovered how to make cells called induced pluripotent cells. They look like and they behave like human embryonic cells, but they've never been near an embryo. They can come from your skin, they can come from your blood. You take those cells, you use these really sophisticated ways of growing them in the lab that kind of deprograms them. And they go back to stem cells that can become anything. So the human embryonic stem cells and the induced pluripotent cells could become blood cells, muscle cells, heart muscle cells, whatever you want them to be. For Parkinson's, we want them to be dopamine nerve cells, right? The ones that are damaged in the brain. And you can produce those very nicely. It took a lot of work to get there, but those can be produced in the lab from either the embryonic stem cells or the induced pluripotent cells. So the induced pluripotent are really interesting because you can choose just one line and you can say, okay, we're going to work with this, we're going to understand it, and we're going to provide this line to multiple different patients. But you can also provide people their own cells. So you can say, all right, we're going to really do a big lift here. And instead of making one line for multiple people, we're going to make everyone their own line. So these are the ones that are called autologous. They're immunologically matched to your own body, your own brain. So you don't need immunosuppression. There's no fear of rejection. It's a lot more of a lift, if you can imagine, to be able to make multiple lines than it is to make one. It's being done though. And I think that's really, really exciting.
I just want to make sure I'm understanding you properly here. So bear with me if you can, and tell me if I'm getting this right. I think what you just said is that you could take a cell from my arm and make a stem cell or make a cell that produces dopamine from that cell. And then if you put that in back into me after you've changed into a dopamine cell, I would not need neurosuppression. I wouldn't need neurosuppression. I would need immunosuppression to make sure that that wasn't rejected by my body and treated like an invader. Whereas if you put a cell from a different from an embryo, that is not mine, obviously.
Right.
You would need to do that immunosuppression. Otherwise, my immune system might attack that cell, and then it would just be destroyed by my body. Is that the
Yeah.
idea?
That's the idea. And then once you get into the actual science of it, it gets really complicated because the brain has some degree of protection from the immune system. And so it may not reject, if you had a kidney transplant and then you're not matched to that kidney transplant and you don't get immunosuppression, it's gone, it's done, it's sort of game over. But for the brain, there is actually a degree of protection. So the cells are not going to be sort of overtly attacked and die, but it does seem from everything that we know that they stand a better chance of surviving well and working well if they're either matched or there's some immunosuppression to protect them in place. So yeah.
Well, okay, so I was close, but not quite exact. Good enough. So I think we should probably tackle an elephant in the room if you're willing to go there with me. And here's the elephant. We get asked questions at the Davis Phinney Foundation quite often, actually, about whether somebody can go to a clinic today, and maybe it's in another country, maybe it's in the United States, but there may be clinics that are offering cell replacement or stem cell therapy today. And I'm wondering if you can say anything about that. Is it reasonable for somebody to do that today, in your opinion?
No, I'll just be really straightforward. So I think there's some blurry lines in there. But there are a lot of clinics that are offering what they call stem cell therapy. They may be using mesenchymal stem cells, like the ones I mentioned at the beginning, that are in real bona fide clinical trials, or they may be using other types of cells that they've developed. And in the US, as long as these cells are only minimally manipulated, there's not as much regulation and as much standardization and as much control around these products as you might like scientifically. So I'll just say, first of all, they're unproven. I think for clinics that are offering treatments for cash, for money, and are not running this in a clinical, under a clinical trial, I would be extremely wary of that. Actually, I wouldn't go near that. The second thing is, there's an argument about, well, it's my right to try. And so I think I just want people to be really cautious because there have been numerous cases in which people have had in which people have had complications. If the cells are not well regulated and you get a contaminant in there, you're really just putting yourself at all sorts of risks. So I would say stay away from those clinics. I would say for people who are interested in looking into the possibility of cell-based therapies, there are more and more going on with research teams who are running these interventions in really strictly controlled environments where you can be sure that what you have has been studied, it's been tested. We know what's there. We know it's not going to cause, or we've tested it for ability to cause tumors. You're testing for infection and all of those terrible complications that we worry so much about.
So it sounds like there are trials ongoing that are maybe using even these mesenchymal cells, and there's certainly other trials that are going on or about to start soon that are using the other options that you were describing earlier, the autologous or the embryonic cells. Autologous, I think is how you say that.
Autologous, yeah.
So
Yeah.
anyway, I'm just wondering if you can say a little bit about the sort of current landscape of trials and the safety and efficacy that we know about already from previous
Absolutely.
trials that have been done in a controlled environment. And I don't know exactly how we want to approach that. Maybe we start with the trials that have already happened and what we know about them, and then talk about the trials that are coming soon.
Yeah, absolutely. So goodness, it's a lot to talk about. If we're just let's just confine it to trials that have come from stem cells, either the human embryonic or the induced pluripotent. And so I'm going to give a couple of examples. And you mentioned previously that there are a couple of very high profile, I guess, publications that just came out in Nature. So I can tell you a little bit about those. And the first study, which came out of Sira, and this is Dr. Jun Takahashi's group, used induced pluripotent cells, a single line, and they had delivered that surgically to the target where the dopamine, where you need to replenish the dopamine input. So they did a surgical delivery in 7 people. And they had used a couple of different doses, no placebo control, which I think is normal in these very early trials because you're really trying to show that you can A, do this, that it's feasible, B, that it's safe, and C, that you're not going to cause so many side effects that no one would ever want it as a treatment. And the Takahashi group certainly achieved that. And so they even published not only safety and tolerability, but in some of the people who participated and received these cells, they could actually see some clinical improvement. Now, I want to be really cautious because you can always say, well, this is placebo effect. It's a very small number of patients, but there it is. And then the second trial was the one that I know a little more about because I was involved in that. And I was at the Vivian Tabar is the neurosurgeon who's in New York who operated to provide surgical delivery, both sides of the brain in 12 patients. So I was the neurologist and senior author on that paper. And what we published was 18 months of following these patients. The cells are like the ones in the Takahashi trial. These are dopamine nerve cells that have come, you've grown them out of stem cells. But whereas June's cells were inducible pluripotent cells, ours came from human embryonic stem cells, and this was a line that's been grown since the 1980s, basically. So Vivian managed to safely operate on 12 participants. The first five got a lower dose, and then the following seven people got a higher dose. And this actually turned into an international study as the Canadians came into play. And as we followed people, again, this is a one size fits all. So everyone got some immunosuppression for 12 months. But we followed them and published at 18 months. So they had come off their immunosuppression. We could actually see evidence on scans that the cells had survived, even when we withdrew the immunosuppression. So it seemed like once they're in, they can hang in there. And the second thing was that exactly like the Takahashi publication, some of our participants actually seem to improve. So it's less off time, it's having less severe offs. There were a whole number of measures that we had looked at. So again, no placebo control, very small numbers, but I think there's enough optimism here to go forward. And the trial that I was involved in was a phase 1 trial. It's actually going to go ahead. That was just 12 patients, but the next aim is 102 people with Parkinson's in a phase 3 trial. So I think this is just going very quickly now. It's really exciting, and really looking forward to seeing those results. But those are not the only ones. And there are a number of groups across the world who are, hot in pursuit of finding a cell-based therapy. So there's another trial that we're involved in now. We just signed up at UC Irvine, again, using cells from a different human embryonic stem cell line. And then the interesting one where we're also involved, Dr. Nick Philippe in my group is the site PI for this. But this is a study funded out of a company called Aspen to take people's own cells. So they are actually taking people's own cells, making their own stem cells, making their own dopamine neurons, injecting them surgically. So I just can't wait to see what comes out of that, because as you mentioned before, this doesn't need immunosuppression. So I think this is a really, really exciting area. And there are other groups who are starting to do this. So it's fast moving.
Yeah, well, it's exciting to me as a person with Parkinson's, knowing so many people with Parkinson's, also that I know so many people with Parkinson's. I'm just really excited to see what comes of this. And I'm actually, I want to draw attention to one thing about the paper that you were an author on. I was really interested in the images of that proved that the cells had survived, and I'm wondering if you can comment about that a little bit, because I thought it was really remarkable, having seen my own DAT scan results, to see those results. It was, first of all, it's beautiful, and second of all, it was interesting from a data perspective. So can you say a little bit about that?
Yeah, and I'll just preface it by saying that the first time we saw those scans, half the room were laughing and half the room were crying because it was such an emotional thing. So you're starting off with scans. These were fluorodopa PET scans. The fluorodopa lights up like two little light bulbs on each side of the brain. But if you have Parkinson's, of course, it doesn't light up as much because there aren't as many dopamine neurons picking up the fluorodopa. So you start off with these scans that look very typical of Parkinson's disease. And then at 12 months, that was when we first repeated the scans. And then at 18 months again, you start seeing it's like filling in of where you want your dopamine cells to be, or where you want your dopamine to be delivered. So it's visually very striking when you look at the quantitation, you look at the actual amount, it seems convincing. And so I think the big thing now is, we've got to follow that over time. So we're looking now at 24 months. We're going to follow people for 5 years total. We hope to follow them for a lifetime.
I'd like to close this conversation by just asking you if there's anything else you think we didn't talk about that we should have talked about, or if there's anything else you just want to share with our audience.
Yeah, I think a lot of the things that I'm very obviously very excited about in the cell therapy world is really going after the dopamine part of the system. And I just want to highlight, I realize full well that for a lot of people with Parkinson's, the dopamine is part of it, but there's so much more, and I think we very much realize that. And so it's a super, super exciting field right now. And I think we just have to keep our eyes open for opportunities to not only look at the dopamine part of the system, but also outside of that. And I think that's where the gene therapy field maybe provide a little more flexibility. And it's possible, like I mentioned at the beginning, that down the line, we're actually going to be looking at some combination of the two approaches.
Yeah, great. Well, thanks for sort of bringing us back around and tying the two threads together again. That's very it's very helpful. So, uh, thanks so much, Dr. Henchcliffe, for sharing some time with me today. I really appreciate it, and I hope maybe we can talk with you again sometime in the future as more information comes to light about these exciting trials.
And I hope I'll have good news for you.
Well, I hope so too. Thanks again.
All right, thank you.